Here we go. Merci. Okay, so first, an introduction on metabolomics and mass spectrometry before we discuss our joint project with Fabien that happened over the last months. So what is metabolomics? Metabolomics is the science of analyzing and identifying small molecules, metabolites. This is done with a technique called mass spectrometry that ionize the molecule and that can measure their mass and deduce some of their structural properties. So mass spectrometry is used not only in metabolomics, but also in proteomics to analyze proteins, and is applied on any type of biomass, plants, clinical applications, and so on. But nowadays, the instruments are so performant that we are generating an enormous amount of data. And so the process of interpreting the results is actually a field that is very active. And of course, it is also using some more classical approaches. But my main message is that even though we can see a lot of metabolites in just a few minutes, most of these molecules are unknown. We cannot annotate or identify them easily. And one way we can resolve this to some extent is to use the fragmentation process. So the molecule is isolated and then fragmented. From that, we obtain a fragmentation spectra that indicates some of the proper structural properties. But the amount of reference spectra that we can compare against is really limited. So in practice, only a very little amount of the information is interpreted. And this is an issue because if we want to understand what's happening in the biological system, then we need to identify most of the molecules. And in the last, let's say, 10 years, a little bit less, some computational methods completely changed this game. And more recently, thanks to machine learning, we have methods that can annotate most of the signals, at least in a partial manner. Okay, I will just continue. So some of these methods I contributed to were developed by the Boker lab in the Jena University. And they built a set of tools that are designed to resolve some of the key steps in the identification process. And so my task was to give a lot of feedback and to test the method and check on the evaluation. And clearly, this tool is now capable of identifying most of the molecular formula, so the atomic composition. We can now also identify known molecules like caffeine. And this is something that is relatively new, predict the chemical class. So we don't know the exact structure, but we can tell that this signal belongs to this chemical class of triterpenoids or whatever. So a quick summary of the process. We use the mass spectrometry data that is processed to detect the signals. And then we use a fragmentation tree analysis to decompose the spectrum into bricks. And from those elemental submolecular formulas, the method used support vector machine to predict chemical properties. So it was trained to recognize some spectral features with a molecular property. And that is used to also search into database of known molecules and coupled with a neural network to associate the fingerprint profile with some chemical classes. And so this was a very active field of research. And I don't have a slide for this, but in 2021, we contributed to a competition, international competition. And using these tools, we obtained the best performance. And for the first time, they were better than the human expert curation. So that was really like a milestone in the field in what we can do in an automated fashion. Okay. So now switching to the KGAI-agent, and this project aimed at bringing some large language model capabilities into metabolomics. So let's start from the context of this study. Prior to joining the CNRS in July, I was at the University of Geneva working with Professor Jean-Luc Wolfender, and I contributed to a project that aimed at exploring one of the largest plant collections in the world from the laboratory Pierre Fabre. And the first task of his laboratory was to design an open knowledge database for all the molecules that were discovered in plants. Because with such a database, we can directly tell, okay, this species should have these molecules. So that was the foundational knowledge. Then those extracts were analyzed by mass spectrometry. So one of my tasks was to run the experiment on the mass spectrometer, and we use some advanced acquisition method. And also the tools I presented to annotate or identify in partial manner those molecules. But also, we collaborated with teams that search for antibiotic activities, pro-metabolic activities. So we have a multi-layer heterogeneous data set with the mass spectrometry data, with biological activities, with knowledge data, very complex and never seen at that scale in our field. So the solution we proposed was to use an experimental knowledge graph applied to the different components of our study. And in a way, this works by having an ontology that includes both the experimental data, so the stuff that we do on the bench, in the lab, but also the literature information and any other information relevant to the study. And that is also interfaced with public resources, so we could query and mine information both in our data set and into the public domain. Okay. So as you know really well, with such knowledge graph infrastructure, one can run some queries to do data mining, and this is really interesting for me with my background in chemistry because we are totally foreigner to these technologies. And the schema used for the representation has a very specific description of the different triples and entities, so mass spectrometry, the molecules identifier, and a lot of specific information about chemistry. But now we have this resource that we want to mine. But what we observed is that, I will switch to this, is that the experts in our consortium that have this deep expertise about mass spectrometry or the biological properties, they don't know how to program in SPARQL. They cannot run requests easily. So there was this dialogue that between the experts in SPARQL with the other experts, and it was not efficient and also a bit frustrating because this is not necessarily fun to write requests for someone else and also have to wait. So our idea was to, instead of training the biological experts to become experts in SPARQL, could we use a large language model to automate some of this process? And this model would translate the question and potentially run the request with the server, the SPARQL endpoint, provide some level of interpretation and an answer in a natural language. And I did some experiment was February 2023, at the beginning of ChatGPT. And what I found is that by providing the sufficient amount of information to the conversational agent, it was able to more or less achieve some decent SPARQL requests. So that was the first indication that there was a way. But there are also limitations. So of course, ChatGPT, as you know, it's made by OpenAI. It's not an open model. We don't know exactly the training set, but some of this information are known. So it's probably not up to date as of today, but it has a knowledge cutoff. So it doesn't know anything about our experimental data, obviously. It doesn't understand what it is without proper instructions. And also there is some variability in the results. So for scientific application in terms of like applied science. This is a bit of a challenge, and some limited capacity to process the information. And soon after, there was the release of the OpenAI application programming interface. So it means that someone can bring large language model capabilities into a Python code, for example, in just few lines of code. So we can call the agent and we can define the different parameters about its task and so on. Okay. And as I said, now we can bring this into existing environments, and we can also combine, or we can give it some instructions that are engineered in such a way that it will achieve its task in an efficient manner. And so you are probably familiar with the concept of agents. So agents are programs that used a large language model along with some instructions and external resources, for example, the New York Times articles, Wikipedia, Wolfram for mathematics, and will use these specific tools in order to respond to some questions relevant to the domain. And what was observed is that by augmenting the large language models with such tools, they were able to have new capabilities that they couldn't answer just from the large language model. And here again, the community in just less than a year released dedicated libraries to engineer these large language models and design such agents. Okay. But this field of large language model is very active for many years. And there is this group in Princeton that published this article about ReACT. So ReACT is for reasoning and acting with large language model. And their experiment showed that if the large language model is tasked to respond in one answer, a one-shot interaction, it will fail for very complex questions. So the question is very complex just by itself, but trying just a single-shot interaction or asking the model to have a reasoning step or a single or even a chain of acting steps did not achieve the result. However, when they engineered a more complex chain, the ReACT chain, where the LLM was tasked with some first thinking about the problem, deciding to act, making some observation about it, and does this in an iterative manner, it was able to answer very complex questions that were not accessible before. So this is quite interesting because we can imagine having like custom chains for some tasks. And this was the inspiration of this project that started during, before July, with a student from the MIT that joined with the support of MIT France, MIT Singer. And also the fact that I didn't realize this initially, but the Sophia Antipolis is a major hotspot for knowledge graph. And for me, with my very basic experiments, that was an incredible opportunity to collaborate with experts on this field. And so we brainstormed together and said, okay, we can probably adapt an AI agent to mine our knowledge graph that was designed for metabolomics. And some of the key postulates are that those general models should be able to learn and extract information without specific training or fine-tuning, given the correct information, but also that the knowledge graph could compensate the hallucination problem with LLM. So LLM will fabricate information when they try to answer specific facts, but here we connect it to the knowledge graph. It might just recover the information and ground it. And since we can engineer it, we can use experts' knowledge to make sure that some tasks that are challenging will be resolved with proper instructions. And so we try to prototype this. The key vision is that we want to have this LLM AI agent connected to our experimental knowledge graph. And the way we achieve this is by designing the agent in such a way that it will first collect all the information needed to answer the question, at least getting facts relevant to the questions from our knowledge graph. So we have the server endpoint. It can query, it has a tool to query the endpoint, retrieve the various identifiers, run some additional code, and eventually succeed and interpret and provide a result to the user. And so another view of it is in this manner. So this is a conversational agent chatbot. So the user can ask a question to the LLM that plays the role of like a maestro, and will decide based on its information. So his role is to answer on the knowledge graph using a set of tools. So the tools are classes that can achieve some tasks. And so based on the tool definition, the agent will decide to use some tools to get information on the species, on the chemical structure, and so on. So this is really like a core component of its behavior. But we also have a chain. So the chain is represented by the numbers here. So it will retrieve information, then it will get some information on the schema, the knowledge graph representation, eventually run the SPARQL query, return the result to the user as a natural language interaction. Okay, so we made a prototype in a few months, and I will show quickly some success and some limitations. So this question is very chemically oriented, so I would translate it an additional layer. So here it asks how many molecules were detected in 2 different experimental conditions, so positive and negative ionization mode, using a tool that was 2 different tools that were used for the annotation of the molecule by looking at their identifier. So it's encoding the structure. So we are asking our large language model to respond to this very specialized question. And what we found is that the agent was capable. I think I'm missing a slide. Okay. So I will comment on this, missing a slide here. But essentially, this question was correctly answered by the agent. This other example, so sorry for this mishap, the other question here asks even more specific question. And we found that, again, after a few minutes of runtime, it was capable of providing the natural language response and also some degree of explainability. So we have the SPARQL query that was proposed and external links to the database, so one can directly look up for the results that are discussed. So in other cases, it works to some extent, but we are reaching the limits of the model, the implementation at least, is that if the amount of information that we need to extract, so this question wants to get a lot of information at the dataset scale, then the response from the SPARQL request is massive and it cannot be run by the agent itself because large language models have a context limit. And so here the solution is that one needs to run this request offline to get the result. So it is still functional in a way. But in other cases, it doesn't work at the moment. So this is the interaction. So we can see each of the steps being processed. So it decides on using this tool and that tool and so on, and eventually running the request. And here it wasn't able to get the results because the question asked for information that are not in the knowledge graph. So this is not easy for someone that is not familiar with the knowledge graph composition. And I presume it will happen quite often, depending on the audience using the tool. But these are some of the limitations. We are still restricted to the perimeter of the knowledge graph at this stage. Okay, so to conclude this presentation, this KGAI-agent is something that is very interesting for metabolomics because it will empower people that are not chemoinformatician, bioinformatician, to do some advanced data mining. So in terms of like democratizing access to advanced capabilities. This is quite interesting. But this is also the early stage. So there is likely a lot of improvement that can be done by some tweaking on the prompt engineering level with feedback from experts, both on the knowledge graph side and the mass spec side. And our goal is to have some, both a web app that can be deployed easily, but also a dedicated library so people can develop in different directions. And finally, some perspective. At the time, we used GPT 3.5, then quickly GPT-4 that perform way better for the prompt engineering part. There will be a new development in the near future, so that's going to be interesting how we can leverage both this OpenAI model, but we can likely leverage other more open models in the near future. And some of the not the dream, but because we can likely be there relatively soon, is to have more than one interaction. At the moment, the question is asked, the agent is performing the different steps, getting an answer. We would like to have some follow-up conversation where there could be some feedback about how to resolve some of the challenges or to be more specific. And so this is just for metabolomics, small molecules, but this is obviously the direction that all of the other omics will take. So genomics, proteomics, they have big data, complex data, heterogeneous expertise in the research groups. So such LLM-based and then let's say hybrid approaches with knowledge graph are definitely the future, in my opinion, for our field and others. So it's an interesting time, and it's going really fast. So that's the end of my presentation. So a few acknowledgements, the funding agencies, of course. Emma, brilliant student from the MIT. So it was a collaboration with Fabien, but also Catherine, Franck, and Pierre. And I hope there will be many more collaboration in the near future. And initially, the knowledge graph was developed with our colleagues in Switzerland, so the Swiss Bioinformatics Institute, University of Geneva, and the professor, the leader of the group in chemistry. And that's it. Thank you very much.